In order for us to understand how recombinant vaccines work, we must know how recombinant DNA works and who thought of the idea of genetic engineering sequence of DNA. Also, we must know a little history about the vaccines and how they got so popular. Let's go through a timeline of the discovery of DNA. So we all know that the structure of DNA was discovered by Rosalind Franklin, James Watson, and Francis Crick. Even after the discovery of double helix, there was many discoveries that were associated with DNA, like semi-conservative DNA replication, successful gene therapy, PCR invention, and there are still much more discoveries to be made. However, before all of these discoveries mentioned, the vaccines were discovered in 1798. Edward Jenner, an English county physician, reported success in providing protection from smallpox by scratching the skin with a pin that held scrapings of cowpox. Jenner did his first experiment on a young boy, scratching the boy's skin with a pin that contained cowpox. After 6 weeks, Jenner repeated the process and realized that the child did not have small sore that the smallpox causes. This is how the idea of vaccines were created. Moreover, another discovery that will be focused on is the development of recombinant technology. How did it all start? On the fall of 1972, there was no such thing as genetic engineering. However, in the early 1970s, scientists from University of California, San Francisco, Herbert Boyer, was working on a lab where he isolated an enzyme that cut DNA at a specific location.
At the same time, Stanley Cohen's Stanford lab was working on methods for introducing small circular pieces of DNA called plasmids in a bacteria. This acts as a Xerox machine, copying genes each time the microbes divide. With that being said, in 1972, a conference in Hawaii gathered both of the scientists, where they presented their work and realized that if they combined their technology, um, they have a remarkable tool. Within months, their lab had jointly proved the possibilities of gene cloning. This is how recombinant DNA were produced. Now, with that being said, recombinant vaccine are vaccines that are produced from recombinant DNA.
The technique or methodology is called recombinant DNA, which is a plasmid made of DNA is removed from a bacterial cell. A restriction enzyme cuts the plasmid DNA open, leaving sticky ends, and the insulin gene with complementary sticky ends is added to create a recombinant DNA. And after the recombinant DNA is made, it gets introduced to a yeast cell. These are cells that are used to rapidly increase the host cell, and this allows for the recombinant DNA to create the recombinant vaccine. Using gene splicing technology, vaccines have been produced. Gene splicing is the joining of separate ends. It's the joining of almost any nucleotide sequence to create a new gene product. Scientists join these snippets in a circular piece of DNA called a plasmid. Scientists use a complex process to clone genes from an organism's DNA. However, in decades of scientific research, most genes already exist in a plasmid stored in a lab somewhere. Gene segments are cut out of the original DNA and joined to make a new gene. Then, researchers check the new sequence to make sure that its position and orientation in the DNA molecule are correct.
So plasmids and the restriction enzymes were used to make recombinant vaccines. We can see this in an example of how hepatitis B was produced. First, the bacteria, which contains plasmid, is going to be cut with a specific restriction enzyme. While that is occurring, hepatitis B antigen producing gene also gets cut from the virus DNA. The recombinant DNA that has been made is placed into the yeast cell because yeast cells are the type of cells that are used to rapidly increase the host cell, and that is placed into a fermentation tank that allows them to multiply and produce more hepatitis B antigens. Thus, the recombinant yeast cell is placed inside the tank, and the recombinant vaccines are made. Recombinant vaccines were the first vaccines that were used for hepatitis B on humans. This relates to the economy as Canada has the GMP certified facilities to produce recombinant proteins, vaccines, and humanized monoclonal antibodies. Also, Canada has the largest per capita number of biotechnology companies. It ranks third in revenue after the US and the UK, generating 1.9 billion per year.
There are some negative side effects that come along with recombinant vaccines, and some side effects which may be serious, while some side effects go away as the body adjusts to the medicine. So some side effects that are included are irritation at the spot where the shot was given, sweating, weakness, tiredness, muscle aches, and nausea.
This is very helpful to our society because it reduces serious diseases from specific type of recombinant vaccines, which make individuals be more resistant to the disease.